Our next guest is going to come on is Scott Harper. And I know Scott Harper is he's kind of I kind of call him like a celebrity a little bit, because he seems to draw a lot of positive energy and attention from the FSHD Society. It's pretty remarkable what this gentleman has been doing for our group, and he joins us now. Hello, Scott Harper. How are you doing, sir?
I'm doing well. How are you?
I'm doing great. I'm doing
Good to see you again.
now. Yeah, you too, man. You are, and I was kind of teasing it. Maybe you kind of heard it a little bit. You're kind of a superstar in my mind when it comes to the FSHD community. Your name comes up and people's eyes light up and like, oh yeah, Scott Harper. He's doing so many great things, and I'm so fortunate to have you on again. And right off the bat, kind of kick it going right behind Mark, which is a tough act to follow. But I know you can do it. So we got.
I followed Mark before.
Have you? Okay, good. So this is old hat for you. Okay. So tell us a little bit about your research there in Ohio State.
Yeah. So we're doing gene therapy.
Okay.
And so let me distinguish gene therapy from other more typical, what we think of like drug therapies. The idea with gene therapy is that we can deliver, in our case, we're trying to deliver a piece of DNA that into muscle cells that can produce another piece of nucleic acid called an RNA. And this RNA we have designed to operate to knock down the DUX4 gene. And so you probably know, and your listeners hopefully know, that DUX4 is this gene that turns on in muscle of FSHD people with FSHD and it wreaks havoc, right? So the idea is that we want to reduce DUX4 in muscles. And so our approach using gene therapy attempts to accomplish that, and actually works really, really well. We've tested it in several mouse models now, and human cells that have been kindly donated from patients with FSHD. And the strategy works very well to knock down DUX4 and protect cells from dying. And so to distinguish gene therapy versus like a drug therapy, the intention of gene therapy is to allow a one-time treatment. So if we do this right, we can deliver this therapy. It will deliver our DNA, which makes the RNA, which knocks down DUX4, to muscle cells, and it will reduce DUX4 in all those cells, essentially permanently. Now, there are some caveats to that. Gene therapy is still relatively new. It's been around many decades, but there are only a few approved products right now. So we don't really know in humans how long a gene therapy will last. But so far it looks like at least a decade, and probably theoretically longer. So that's the idea. Our ultimate goal is single treatment to deliver a nucleic acid that can reduce DUX4 and hopefully be done with it.
That is amazing. And the fact that, even if you said it lasted a decade, I'd be like, all right, then sign me up. I'll go back again. Is that the essence of, is that kind of a worst case scenario in a way? People might have to go back for a second treatment or third, or make it just a lifelong treatment.
Sure.
Also a possibility.
That's a great, so this is where the field's going now. So the essence of gene therapy, that's a great question. The essence of gene therapy is we use what's called viral vectors. So these are modified viruses. The ones that we use are called Adeno-Associated Viruses, or AAV. You might have seen that a lot in the news. AAV are these viruses that they naturally occur. They're in the wild. There are lots of different types, and they don't cause any disease in humans. But what we can do is these vectors, or these viruses, have spent millions of years evolving ways to deliver genes into muscle. A lot of them really like to infect muscle. So what we can do is we can take out the virus's genes, and we can replace the virus's genes with the the piece of DNA that I told you about that can knock down DUX4. We can grow that virus in a dish in the lab, and then deliver it back to. In our case, so far, what we've done is mice, but we're working to get to test this in humans. So I'm getting to your question here. Just bear with me. So we use these viruses then to deliver this therapy. And as you can imagine, I mean, when you think about you get infected with the virus, we've got this pandemic going on right now, and what's the big push? It's to try to develop a vaccine, right? So we can fight off this virus. And so essentially what happens is when you deliver even a gene therapy virus like we're using, you become immunized to it. Okay, so it makes it difficult then to come back in and deliver it again.
Okay.
Okay, because your immune system will recognize it as something foreign. It's seen it once, you can get away with one treatment, but if it sees it again, then you've got antibodies now to that, right? So the gene therapy field now, it's actually, in my opinion, it's a good problem to have, because of what it means is that the gene therapy field has matured to the point now where we actually start thinking about, can we redose later on? I mean, it's an important development in our field. So gene therapy, to be clear, we have, and when I say we, I mean our field, the gene therapy field. There are approved therapies for blindness and for spinal muscular atrophy on the market right now. They've been FDA approved, using the same AAV vectors that we want to use for FSHD. So this is not science fiction. This is in the realm of possibility. And what we need to do, the next step to get to your question is, if we want to have the ability to redose later on, to give somebody a booster shot, say, we need to think about how to manage the immune response that you would get to that first administration of the virus. The field is not quite there yet, but there are some pretty cool things going on to try to address that problem. So I would say that right now, it's not possible to do, but I think it's going to be. It's just not. It's not ready for prime time yet.
Wow. So the virus is a good thing. It is the delivery.
Exactly.
To help us. Wow. That is phenomenal. I think that's kind of my small little tiny blessing of this whole COVID stuff, is that you hear about all that science, and then when I talk to the researchers such as yourself and the doctors and whatnot, that it kind of starts to come together in my mind a little bit. Oh yeah, RNA. Oh, I've heard that before. And it starts to connect the dots in my not so advanced mind when it comes to the research side of FSHD. So people such as yourself to help diagnose it and then break it apart and help me understand it helps me then share the news with people that don't even know of FSHD and what their money can do, and what their donations can do and how it can help. How can people help the most in your mind, Scott? Is it clinical trials? Is it volunteering themselves, donations, all of it, in your opinion?
Yeah. So I mean, you pretty much touched on all the major points here. So just to give you a sense of where we are and where we've been in our program. I've been working on FSHD for about 13 years, and when I started in the field, this gene I mentioned, DUX4, DUX4. It was kind of just lingering around the periphery. Nobody really understood that DUX4 was the gene. It was the thing that we wanted to tackle. So it took some time in our field to come to some consensus on that this is our gene target. There were no models, so to make a therapy, you need to have some sort of cell and animal models, right? We didn't have any because we didn't even really understand the mechanism behind the disease. And as you might imagine, don't understand what causes it, don't have models, can't really develop treatments. Right. And so my lab's been focused on making models and trying to use those models to test therapies. And I already mentioned we're doing gene therapy. So the very first, when I first started in the field years ago, my lab just opened, and I was looking for some funding. We had this idea for a new model, and I emailed Dan Perez, who is the CEO of the FSHD Society. Actually, it was the FSH Society at the time. And I said, hey, Dan, I got this idea. When's your next grant cycle? And he said, well, you just missed it, but why don't you send me an application, send me something, and I'll put it to the scientific advisory committee. Anyway, make a long story short, it was just a small grant, $10,000, but it got us going and it helped us make the first model that we used to develop our gene therapy. So that was years ago. Since then, we've been able to get NIH bigger grants from the National Institutes of Health to make other models and develop other therapies. And we've gotten our program now to the point where, I mean, it works. We're convinced it works really well, but it becomes expensive to move it to the next stage, which is the clinical trial. And so that sort of segues into another point that you made, which is that now our program is matured to the point where we know it works. In mice so far, everything we've seen, we've done some extensive toxicology studies that we haven't published yet, but everything looks great. I mean, the mice have looked fantastic. It seems to be very safe. And so now we're to the point where we've partnered with a private company. We'll make an announcement more on that sometime soon. And we're aggressively pursuing getting this to the clinic. And so we're at a point now where, before we needed these small grants to start developing our program and donations to support the FSH Society. And now we're at a point where we're going to be looking for volunteers to help with testing to see if this works in the clinic.
So what does that timeline you think look like from this day's point of view? I know that can very easily change, but a hopeful timeline, when it gets to trial and et cetera.
Okay. I've been talking about this program for about 3 years now. We've advanced it sort of under the radar, especially on the safety aspects of it. And like I said, we haven't published any of that work. But I've been trying to find gene therapy, especially, gets very expensive to take to the clinic, and it's not something that you can fund easily with NIH money or foundation money. So at some point, it requires some private funding from corporations or biotech companies. And so we've been for about 3 years now trying to find the right partner, and we finally have, thankfully. I mean, we have an aggressive plan to move forward now. But I've been telling people, oh, we're going to be in the clinic soon, thinking that the business part of the deals were going to get done more quickly than they did. Mm-hmm. So now that we have a deal in place, and we have some partners that I think know what they're doing and are going to aggressively push this, realistically, I think in a year we can be in the clinic and probably have data within 2 years. But that's with the caveat that everything moves along the way we expect, and you never know what happens. Like we had a pandemic that slowed research down. So I'm hopeful, based on what I'm seeing now, that we're going to be able to move quickly on this now that we have this strong proof of concept data in animals and safety data and the right partner. And I'm excited about trying to get this moving quickly. I mean, we're serious about taking this to the clinic as quickly as we can.
Mm-hmm. Oh yeah, I mean, I have no doubt in my mind. I mean, I can always hear the passion that you have for the FSHD community and your research with it, and that it's more than science, it's people. I think if
absolutely.
anybody has it and has that knowledge and understanding, it's definitely you. And I think that's also why people draw to what you have to say about your research, is because they feel that, and maybe I know I'm speaking for the whole community here, granted, but I would honestly say that we know that you're in our corner. This is someone that is fighting for us, and it feels like you might even be a little bit, emotionally attached to your research. I mean, am I accurate in that? This is just more
absolutely, yeah.
Right.
I mean, not only because I've been working on it for so long, but
right.
Over the years, Dan Perez, like I said, he kickstarted my lab early on, right, when he didn't have to do that. And I've met over the years, I've met so many people who are living and struggling with this disease that have become friends.
Mhm.
And I desperately want to see something, even if it's not ours. I think ours is great, but I mean, anything to help the community, we're willing to do.
Final question for you. What is the
Yeah.
importance of like a donation type event like this to your research or to the community as a whole from your perspective? Like a Giving Tuesday type day like this.
Yeah, like I said, the FSHD or FSH Society at the time, I still have to get used to that that switch. Kickstarted my lab, and technology moves along so quickly. You never know what the next. Whoa! I just lost my camera there.
We got you. I see it.
Okay. You never know what's going to be the
Yeah.
next big thing, and to be able to have research starts usually with foundations like the FSHD Society funding early ideas that then you can use as a seed to get larger grants, and that it's absolutely critical to have funding from the FSHD Society and other foundations to kickstart those new ideas, because it's hard to get them funded any other way. It's hard to get them off the ground.
Right. The fuel. It's the fuel you need.
Absolutely.
Money and participation from patients and the support. Appreciate your time today, Scott. Thanks for joining us. I know you're a busy guy. I want to get you back to work as quickly as possible. I don't want to keep too much of your time, even though it's awesome talking with you. Believe me, it's a great pleasure to have you here.
Well, I appreciate it. And if I can just say also before I go, once the pandemic's over, anybody who wants to come visit the lab, it's an open door. All we need is a quick email saying, hey, we'd like to come visit and meet everybody and see the lab. We do that all the time. It's just been tough because of COVID-19, but I just want to put that invitation out there.
Well, and since you kind of brought that up, if people wanted to find out more about yourself or the research you're doing, of course, or emailing you, how can they do that? What are the best contact ways?
Yeah. I would say just directly my email address at work, which is scott.harper@nationwidechildren's, all one word, .org.
Perfect. Excellent. I'm glad you could share that info out for us. We're gonna have to pay a visit after this pandemic thing is over.
Absolutely. Love to see it.
Thanks, Scott. Have a good day, sir.
Good to see you. Thank you.